Okay, welcome to lecture 3 of chapter 9. In this lecture, we're going to talk about anaerobic versus aerobic conditions, specifically when cell respiration happens and when cell respiration doesn't happen. Cell respiration is an aerobic process, meaning it requires oxygen. And if you recall from last lecture, oxygen is the final electron acceptor of the cell respiration or chemiosmotic pathway, meaning oxygen is going to be inhaled, it's going to diffuse through the tissues and bloodstream of your body, and will end up accepting low energy electrons from the electron transport chain after they actively pump the hydrogen ions from the matrix or low H+ ion concentration side across the mitochondrial inner membrane, the cristae, to the high hydrogen ion concentration side. But once those low energy electrons leave the electron transport chain and have already, and have given off all their energy, they're going to be accepted by the final electron acceptor. The final electron acceptor in cell respiration is oxygen. And so when oxygen isn't present and we have an anaerobic environment, then cell respiration cannot happen because you cannot remove those low energy electrons without oxygen. Okay, and so now we're going to shift gears and we're going to talk about anaerobic environments and why cell respiration can't happen within the anaerobic environment and what happens within an anaerobic environment. So we already talked a little bit about why cell respiration can't happen within an anaerobic environment, because it doesn't contain oxygen, and so oxygen isn't present for the final electron acceptor, and so it won't remove those hydrogen ions or won't remove those low energy electrons. And so in your body there is sometimes no oxygen to use in order to remove those low energy electrons from the electron transport chain. How many of you, and I know you can't really answer me right now, but how many of you have experienced lactic acid fermentation? Meaning fall sports are over. Maybe you were out for a fall sport, maybe you weren't, but winter sports just started, and so maybe you are trying out for the basketball team, or maybe you went out for football or whatever, and over the course of the first few practices you found yourself incredibly sore the next day. That is the result of lactic acid fermentation. Okay, the reason you're sore is because your muscle cells ran out of oxygen and therefore couldn't continue to produce ATP through that chemiosmotic pathway. And because you no longer had oxygen available within your cells, the cell no longer could do cell respiration and had to switch gears to lactic acid fermentation. Okay, fermentation is a process of producing ATP without oxygen, in the absence of oxygen. And I know you say, well, I never stop breathing, so how is the cell out of oxygen? And the answer is, yes, you as an organism breathe, but you don't diffuse oxygen into your cells fast enough. Your oxygen that you diffuse in is basically used faster than you can get it into the body. Now, when you're going through some form of vigorous exercise or vigorous sport or whatever, the oxygen has to diffuse faster, which is why your respiration increases. Okay, the longer or faster you run, typically the harder you breathe, and that's because the body is responding to the stimulus of you increasing the exercise level, and so you are trying to diffuse oxygen in at a faster rate. But the cells that are producing large amounts of ATP have to use that oxygen basically faster than the cell normally would, and so you switch gears to lactic acid fermentation. And there's two different types. We do not do alcoholic fermentation. We do lactic acid fermentation, but alcoholic fermentation is just as common on the planet. Alcoholic fermentation is important within the beer or wine industry. Okay, you're utilizing an anaerobic environment to produce alcohol as a byproduct of this fermentation process. We, on the other hand, produce lactic acid as a byproduct of this fermentation process. So a little bit more about alcoholic fermentation. We are basically talking about yeast. Okay, and yeast are single-celled eukaryotes, meaning they contain a nucleus, but yeast cells don't produce lactic acid. They produce alcohol in an anaerobic environment. And so they're going to go through glycolysis because glycolysis is an anaerobic condition, okay, or an anaerobic process. And it makes sense because alcohol, or glycolysis rather, doesn't require oxygen as a final electron acceptor anywhere within that glycolytic pathway. Yeast are going to convert glucose into pyruvic acid just like we do, and they're going to produce NADH like we do, but because they don't have oxygen available, you aren't going to send, or they aren't going to send that pyruvic acid to the prep step, Krebs cycle, and NADH. Back that up. They're not going to send the pyruvic acid into the prep step and then ultimately into the Krebs cycle because there is no oxygen available at the electron transport chain, and so that entire pathway kind of like halts to a stop. They're going to basically give the hydrogen and electrons back to the pyruvate once they're made, and in a side reaction they're going to produce alcohol, CO2, and NAD+. And the reason that they're basically giving the electrons and hydrogens back to the pyruvate is because they want to regenerate this NAD+ so that glycolysis can continue, because what is ultimately produced through glycolysis? It is a net of 2 ATPs, remember? So even in the absence of oxygen, we can still produce a net of 2 ATPs per glucose, which isn't a lot, but it's some, and it's better than nothing. And as a byproduct, produce alcohol or lactic acid. And again, you see this when you talk about like bread dough rising. Okay, how many of you have had a parent, or maybe you've even done it yourself, make bread dough and leave it on the counter for several hours, and the longer you let the bread dough rise, the bigger it gets. And when you bake the bread, obviously all those carbon dioxide bubbles and alcohol bubbles evaporate, leaving kind of the spongy looking bread behind. Okay, all those little pores in a bread slice or a bread loaf are basically the little pockets of carbon dioxide and alcohol that were produced during the alcoholic fermentation process that is happening while the bread is rising. Lactic acid fermentation, it's the exact same thing. You're going to, in your cell, take glucose, you're going to convert it to pyruvic acid, just like you do normally in the glycolysis process. You're then going to convert the pyruvic acid into lactic acid by regenerating NAD+ from NADH. And the reason we do that is not to produce the lactic acid, but it's a form of quick energy. Again, we can produce 2 ATPs per glucose, which isn't a lot, but it's bonus, it's some, and even in the absence of oxygen, you still can produce ATP, and that's incredibly important when you're undergoing rigorous exercise or rigorous physical activity. Okay? So if we talk about kind of this lactic acid fermentation process, again, glycolysis is an anaerobic condition, and so glycolysis will take glucose and produce it to pyruvic acid or pyruvate. So this portion right here is glycolysis. While glycolysis is happening, you are converting 2 ADPs into 2 ATPs. That is a net gain of 2 ATPs. Okay? That is the result of substrate level phosphorylation that happens during that glycolysis process. As you produce ATP, remember we produced 2 molecules of NADH from 2 molecules of NAD+. Now, think of it like this. NAD+ is going to grab 2 electrons and a hydrogen ion from the 2 G3Ps to produce 2 NADHs. They're going to temporarily hold on to those electrons and hydrogens while you convert the G3P to pyruvate. Well, once you turn it into pyruvate, you essentially give the 2 electrons and a hydrogen ion back to the pyruvate, turning it into lactic acid or lactate. Why is that needed? You need to regenerate NAD+ so that it can go back and hold on to temporarily the electrons and hydrogen ions from another glucose molecule so that you can keep this glycolysis processes happening to ultimately produce 2 ATPs, which, like I said before, is not a lot, but it's some. Okay, it's better than nothing. So here is the last kind of process, or the last diagram of chapter 9. This is the difference between aerobic and anaerobic. Aerobic is with oxygen, anaerobic is without. Glycolysis, remember, is an anaerobic process, which means it will take place without oxygen, but it can take place in the presence of oxygen. It is not dependent on oxygen. It happens regardless. And so glucose, through glycolysis, is going to be converted to pyruvate always, and in the process are going to convert 2 ADPs to 2 ATP. Now, if oxygen isn't present, the pyruvates will branch this way, and if oxygen is present, the pyruvates will branch this way. If oxygen is present, the rest of cell respiration will take place, which means the prep step will happen, Krebs cycle will happen, ultimately the electron transport chain will happen, and we will produce that total of 36 total ATPs. That's a lot. That's best case scenario. Oxygen is present, we produce 36 ATPs. If oxygen isn't present, you're going to temporarily produce the NADH, but you're going to give the hydrogen and electrons back to the pyruvates, turning the pyruvates either into lactic acid or alcohol, depending on what organism you are. If you're a yeast cell, you will produce alcohol. If you're a mammalian cell, you will produce lactic acid. Okay, we will review this thing or these processes in class. Make sure you took good notes. Bring the questions that you have to class, and we will get all of this ironed up. See ya.